Welcome to my YouTube channel. I will start a series of lecture videos titled Basic Microbiological Tests. The first in the series is Biochemical Identification of Escherichia coli. Escherichia coli, hence called E. coli, is a gram-negative non-spore-forming motile rod, and it forms part of the normal flora of the human intestine. When introduced outside the human intestine, for example, the urinary tract, it can cause severe infections and it requires adequate medical attention. The detection of E. coli in environmental samples is an indication of the presence of pathogenic bacteria such as Salmonella or Shigella species. Hence, the rapid identification of E. coli is of public health significance. The biochemical identification of Escherichia coli include indomethyl red vesicrystal citrate utilization tests. These are collectively called INVIC tests. The first in the battery of tests to be discussed is the Indo test. Let's look at the protocol of the Indo test. The pure cultures of the test and control organisms are grown separately in 5 ml culture medium containing the amino acid tryptophan. The most important or usually used medium is trypticase soy broth or trypticase soy agar. The inoculated culture medium is incubated at 37 degrees Celsius for 48 hours, to which 5 drops of COVAX reagent is added and shaken gently. A positive test is indicated by the appearance of a red color or ring in the surface reagent layer above the broth within 1 minute. What is the principle behind the Indo test? The test organism should be able to elaborate an enzyme known as the tryptophanase, which degrades tryptophan in the medium to 3 products. These include indopyruvic acid and ammonia. The indo so produced combines with an active component of the COVAX reagent known as para-dimethyl aminobenzaldehyde. This combines with indo to produce a red colored compound, especially when the pH is 4.4 or below 4.4. What is the application of this Indo test? Specifically, Escherichia coli gives a positive Indo test reaction. Here is a plate showing the Indo test. On the left, the first tube there is the uninoculated tube containing the trypticase soy broth. The middle tube shows the positive Indo test. Here, E. coli gives a positive Indo test in which there is a red color, a red ring in the reagent layer. And the third tube shows a negative Indo test where there is no red color in the reagent layer. This image was obtained from the American Society of Microbiology. There are alternate Indo tests. These include the sulfide Indo motility test, the motility Indo ornithine decarboxylase test, and the Indo spot test. All these are new Indo tests that were develop, and they are rapid tests, particularly the first two, the SIM, sulfide endomotility test, is a multiple test which involves testing for the production of hydrogen sulfide, endo production, and motility in the same tube. The red coloration of the upper part of the medium, when a few drops of Kovacs reagent is added, indicates that indole has been produced. The second alternate indole test is the motility indole ornithine decarboxylase test. Here again, 3 tests are combined in one test. Here you have the motility test, the indole test, and ornithine decarboxylase test. Then the indole spot test is similar to the oxidate test, in that following the growth of the organism, a piece of filter paper is wet with a few drops of Kovacs reagent, and then the organism, a colony of the organism, is smeared on this wet filter paper. There is an immediate development of a red or color on that spot. The second test in the battery of tests for identification of Escherichia coli is the methyl red MR test. The procedure involves inoculating the pure test organism into a 5 ml methyl red prescribed MRVP broth, and this tube is incubated at 37 degrees Celsius for 48 to 72 hours. Control organisms should be included in the tests performed. After incubation, 1 ml of the MRVP broth is transferred into a small serological tube, and 2 drops of methyl red indicator are added. A red coloration of the MRVP broth is a positive MR test, while a yellow color indicates a negative MR test. The principle behind these tests, the MR or methyl red test, is that following the growth of the test organism in the MRVP broth, there is a pH of 4.4 or even below that, and the methyl red indicator changes to the red color in this acid environment. Escherichia coli gives a positive methyl red test. There is a plate showing the methyl red test. This image was obtained from the American Society for Microbiology, ASM micro library. On the left is a tube showing the positive MR test, which E. coli is positive. And on the right is a negative methyl red test. The third in the series of the tests for identification of Escherichia coli is the what is prescribed VP test. To the rest of the MR VP medium, 0.6 ml of 5% alpha naphthol in absolute ethanol, which is actually the Barrett's reagent A, added, followed by 0.2 ml or 4 drops of 40% potassium hydroxide in deionized water, which is actually the Barry's reagent B. The tube should be shaken and the cap loosened or removed and kept. The tube should be kept in a slanted position. The appearance of a red color within 30 minutes to 1 hour indicates a positive VP test. What is the principle behind the prescribed test? Bacteria ferment the glucose present in the MRVP medium through the butanediol pathway, producing an intermediate known as acetion. This acetion combines with, or rather is oxidized in the presence of the potassium hydroxide, and a new product known as diacetyl is produced. This reaction is initialized or catalyzed rather by the alpha naphthol. Alpha naphthol is known to be a color intensifier. Hence, the red color that appears in the positive Voskowska test. The diacetyl reacts with the guanidine group associated with molecules which are contributed by peptone that is formed or that is present in the medium. Here is a plate showing there are two tubes. The tube on the left is a positive VP test or Voskowska test, and this is not Escherichia coli is Voskowska or VP test negative. So it is actually is the tube on the right that is negative. But for an organism like Enterobacter aerogenes, which is VP or Voskowska positive, there is a red coloration of the broth. The fourth test to be conducted in the identification of Escherichia coli is the citrate utilization test. The protocol involves the inoculation of the test organism as well as control organisms on the surface of citrate, Simon citrate agar medium slant by streaking and then stabbing the butt of the medium with a sterile inoculating needle. The inoculated tubes are incubated at 37 degrees Celsius for 48 hours. A positive citrate utilization test is indicated by a blue coloration of the medium. The principle behind this test is that citrate is the sole source of carbon in the medium. And what's the medium? Simon citrate agar medium. So as the test organism grows, an alkaline environment is created, and the pH indicator, which is known as bromothymol blue, changes to blue, which is its color in the alkaline environment. Escherichia coli gives a negative citrate utilization test. Here is a plate showing the uninoculated citrate medium or the Simon citrate medium in slant form. Escherichia coli gives a negative citrate utilization test. So it has the same color, that is the green color, as the uninoculated medium. While Enterobacter aerogenes gives a blue coloration of the medium. So E. coli is negative for the citrate utilization test. Here you have the typical IVIC test results of E. coli. E. coli is Indo-positive, Methyl-right positive, Vos-Pascal negative, and citrate utilization negative. That is the typical IVIC test result. There are other rapid identification systems for Enterobacteriaceae. You have the BBL enterotube 2 system, you have the micro ID system, and then you have the enteropleural system, API 20E. Using the BBL enterotube 2 system, here you have the image which was sourced from the American Society for Microbiology laboratory. Is a tube which consists of 12 components, and various tests are carried out. Here is an example for Escherichia coli. And the expected results, the first tube, the glucose medium, it turns yellow. Gas is not seen, not usually seen. This is because glucose has been fermented. Then also the lysine, the carboxylase medium turns purple, which is positive. Escherichia coli is also positive for ornithine decarboxylase test because the medium turns purple. Hydrogen sulfide is not produced. There is no blackening of the medium in the enterotube or SIM tube. The Indo test is positive, where you have the red ring, which will be seen at the top of the SIM tube after addition of COVAX reagent. There are 5 sugars that are usually used in this test. The BBL enterotube 2 identification system for Escherichia coli. You have adonitol, which is not fermented, so the medium remains red. Lactose is fermented, the medium turns yellow. Arabinose is positive, the medium turns yellow. Sorbitol, it turns yellow. Jucitol is negative. Where the alanine medium remains greenish, that is, E. coli is negative for the phenylalanine decarboxylase test. Urease tests, for the urease test, the urea medium remains yellow, while the citrate medium remains green. It means that E. coli is negative for the citrate utilization test. Thank you for listening. And here are some references for further reading.